Earlier today, 2 suspects were seen fleeing from the crime. I was called in to determine who committed this horrible crime. I have just arrived at the crime scene. The victim was found dead in the kitchen. I will now examine the victim's body to figure out who her attacker is. I have found some hair that the victim must have grabbed from her attacker's head. I will use this along with using the form of DNA fingerprinting to figure out who her attacker was. Here are the 2 suspects found fleeing from the crime scene earlier today. Suspect 1, Suspect 2. I have just arrived back at the lab with the hair samples. I will now place each hair sample in a detergent in order to extract the DNA from the cells. Suspect 2 hair. Suspect 1. And the hair found in the hand. After a few hours, the cells have dissolved, leaving the DNA behind. I will now take the DNA and place them into their own test tubes. Suspect 2. Suspect 1. And the hair sample found from the crime scene. I will now place the restriction enzymes, which cut up the DNA into smaller pieces, into each of the test tubes. I will now shake up the DNA with the restriction enzyme in each test tube, and in a few hours I will add the DNA to the agar gel. After a few hours, the DNA has been successfully cut up by the restriction enzymes. I will now add blue dye to the solution so we can track the DNA movement through the agar gel. The next step is adding the dyed DNA into the wells of the agar gel. The hair sample from the crime scene will go in the first well. Next, in the second well, is suspect 1. And last is suspect 2. I am now going to attach positive and negative charges to the agar gel and place it fully submerged in water in order for the DNA to precipitate through the gel. The positive end will go at the bottom because DNA is negatively charged and will attract to the positive. The negative will be at the top in order for the DNA not to jump out of the agar gel. I am now placing it in water. And I will now turn on the battery. We will now have to wait a few hours. Because this is such a long process, I will be showing you a simulation of what the DNA would look like going through the agar gel. After a few hours, the DNA has moved through the gel. As you can see, the larger particles settled at the top while the smaller particles settled at the bottom. This is because of the smaller particles' abilities to weave through the gel much faster than the larger particles. The sample matched a perfect match with subject 2 because of how its DNA lined up exactly, as you can see on the agar gel.